Meiosis is a two-part cell division process in organisms that sexually reproduce. Before dividing cells enter meiosis, it undergoes a period of growth called interphase. During interphase, the cell increases in mass and preparation for cell division. Interphase is composed of three parts: G1, S phase, and G2. In G1, the cell increases in mass in preparation for cell division. S phase is the period in which DNA is synthesized. In G2, the period after DNA synthesis has occurred but prior to the start of prophase, the cell synthesizes proteins and continues to increase in size. In prophase 1, chromosomes begin to condense and homologs loosely pair. Pair homologs become physically connected to each other along the lengths by a zipper-like protein structure. The state is called synapsis. Crossing over is a genetic rearranging between non-sister chromatids where the exchange of DNA molecules happen. Crossover begins during pairing and synapsis. The chiasma is where crossover occurs. Also, during prophase 1, the disappearance of the nuclear envelope.
Pairs of homologous chromosomes now line up at the metaphase plate side by side. Both of the chromatids of one homolog are attached to the connectochore microtubules from one pole.
Anaphase 1 begins with the two chromosomes of each tetrad separate and start moving towards the opposite poles of the cell as a result of the action of the spindle fibers. Notice that in anaphase 1, the sister chromatids remain attached to their centromeres and move together towards the poles. A key difference between mitosis and meiosis is that sister chromatids remain joined after metaphase in meiosis 1, whereas in mitosis they are separate. As this occurs, the cell begins to elongate.
At the beginning of telophase 1, each half of the cell has a complete haploid set of duplicated chromosomes. Cytokinesis is the division of the cytoplasm which happens simultaneously with telophase 1, forming two daughter cells. In animal cells, like shown, a cleavage furrow forms. In plant cells, however, a cell plate forms. Next, chromosomes decondense and nuclear envelopes form. No chromosome duplication occurs between meiosis 1 and meiosis 2. Now meiosis 2 can begin.
In meiosis, the cell goes directly from telophase 1 to prophase 2 without the interphase. In prophase 2, the nuclear envelope is again dissolved and a spindle is set up again. Prophase 2 is identical to prophase of mitosis except that there's half the amount of chromosomes. Chromosomes are condensed and do not replicate in this phase of meiosis. In metaphase 2, in each of the two daughter cells produced by the first meiotic division, the spindle again draws the chromosomes to the metaphase plate. This time, unlike metaphase 1, the two gonadotropes of each centromere bind to spindle fibers from opposite poles. The chromosomes line up single file at the metaphase plate at the cell center.
During anaphase 2, the sister chromatids of each chromosome separate and move toward opposite poles. As the chromosomes are dragged along by the spindle apparatus, their arms can be seen dragging along behind so that the chromosomes form V shapes. The poles themselves move further apart as cytokinesis begins and the cell elongates. During telophase 2, the chromosomes reach opposite poles, cytokinesis occurs, the two cells produced by meiosis 2 divide to form four haploid daughter cells, and nuclear envelopes form. Meiosis is now complete.